With that, I'm going to hand it over to Peter Meikle.
Thanks, Val. And let me also welcome everyone to this first session. It's my pleasure today to introduce Professor Elaine Holmes. So in Australia, we're very fortunate that we were able to attract Elaine to move from the UK to take up the position as head of the Centre for Computational and Systems Medicine at Murdoch University. So that was about a year ago. Unfortunately, since Elaine's moved over, Western Australia has been pretty much isolated from the rest of Australia. So Elaine hasn't been able to visit anyone, and I don't think many people have been able to get over to Western Australia to visit her. So we hope that will change soon, Elaine, and you'll be able to move more freely around the country. But it might still take a few more weeks at least. Anyway, look, you can see that Elaine has a very distinguished career. She's been awarded numerous prizes, but particularly the prestigious Australian Laureate Fellowship from the ARC here since she arrived just a year ago. And so her current work focuses on progressing research into maternal and infant health, liver and gastrointestinal disease, and metabolic diseases. And today she's has the very topical title, Metabolic Signatures of SARS-CoV-2 Infection. So Elaine, I'll pass over to you.
Oh,
You're
Elaine.
still muted, Elaine.
Yeah.
Is that better? I'm just trying to figure out how to share this screen.
If you go to the bottom of the screen there, Elaine, and
Yeah.
find the share screen button, it's just a couple down from participants and chat. And then you should be able to do that and share the application. So find the PowerPoint.
Oh gosh. There we go. Can I get that onto full screen? Oops, I'm just going to the... Is that working?
Yes.
That's great.
Great. Okay, thank you very much for the invitation to speak today. It's been great in WA. We wouldn't know we had a pandemic of COVID except for we can't go anywhere. It's kind of a bit like Hotel California at the minute, but I'm not complaining because it's sunny and it's beautiful. So today I'm going to talk about metabolic profiling of SARS-CoV-2 infection. And really, all the things I'm interested in, like diabetes, dementia, liver disease, this particular infection seems to stitch all of these things together. And if we just consider COVID, people started off thinking of it as a respiratory infection, a virus that, you had the sneezing, sore throat, whatever, fevers, and the difficulty in breathing. But it's becoming much more apparent now that it's so much more. So if you look at the symptoms people have, it's really wide ranging. So it goes from things like fevers and coughs, but right through to fatigue, acute cardiac injury, nausea, loss of appetite, loss of smell. And you're even seeing this Kawasaki, like the syndrome in children where they have burst blood vessels, particularly in the toes and hands and extremities. So it's got a very wide range of symptoms. And we also know, though, that a lot of different risk factors come into play. If you're older, you tend to be more susceptible if you have cardiovascular disease or cerebrovascular diseases. Sometimes if you have some of your raised cytokines and inflammatory markers, again, more susceptible. But nobody's really been able to piece it together properly yet. But what's perhaps even more worrying is that there's a clear long-term health impact. So if you get over COVID and you recover, you still have to contend with some symptoms. A lot of people are coming back months later still with residual symptoms, be it ventilation issues. People are seeing there's still blood gas diffusion anomalies. There's some evidence of psychological dysfunction, neuromotor dysfunction, persistent lung lesions. and the list goes on. So we're interested in doing two things. One is early diagnosis. More importantly is prognosis of, can you tell early on in the disease how severe somebody's going to be, and perhaps hospitalize them and give them treatment earlier, because there's evidence that the earlier you give somebody oxygen, the better their outcome or prognosis. But then also, can we predict or get any idea at all of which potential long-term symptoms are going to persist, if any? And if you look in the literature, there's quite a lot beginning to come out. We can see things like metabolic profiling has shown ketone bodies and liver dysfunction. So there's a link with the ketone bodies and elevated glucose. There's a definite link to diabetes. We know that you're more susceptible if you start off with diabetes, but the COVID infection, people are starting to publish research saying that it does actually tip people into either type 1 or type 2 diabetes. And there's a lot of discrepancy in the literature, but clearly there's a metabolic angle here. Another of the metabolic profiles we're seeing is disruption of the tryptophan kynurenine pathway, and then very clearly a third level of totally disrupted lipoproteins and lipids. So in our laboratory in Perth, we've got the Australian National Phenome Centre, and the technology is split between NMR spectroscopy and mass spectrometry. I'm going to present a little bit of results from both today. The things we're focusing on mainly in terms of the COVID screening has been the lipoprotein profiling on the NMR, and on targeted amino acids and tryptophan assay analysis. We've also screened for the lipids, but haven't had time to analyse those results fully yet. So we were lucky in WA in that we didn't have much COVID, so much so that it was really, really difficult to get samples. So we started off with a very small sample set, but the sample set we had from Western Australia, which was only based on 17 patients, has since been backed up in the Cambridge data, but I'm going to focus on the WA report today. We're currently importing samples from both Cambridge and Spain because we don't have enough in WA, but all three cohorts so far are coming up with the same results. So just having a quick look at the cohort demographics, we can see that the main comorbidity here, or predisposing factor, seems to be hypertension other than age. And unlike most of the world, because it's a small cohort, we don't see the big male-female discrepancy. We actually had more females in our cohort. But if you look across the symptoms at the bottom, you can see that really wide range of symptoms: fever, cough, shortness of breath, but fatigue is right up there too, and also diarrhea. So the gastrointestinal and the fatigue syndrome as well. So if we consider COVID-19, the clinical progression of this viral infection, you've got the incubation or asymptomatic or mild symptoms phase, and some people stay in that phase and don't progress any further. So this can all be managed out of hospital. At some point, some people will go into interstitial pneumonia, followed by acute respiratory distress and multi-organ failure in some. And you also have this prothrombotic phase, which is the particularly nasty phase towards the end of the disease. And hopefully most people are recovering and going into remission, but obviously there's some mortality, quite a high mortality associated with the virus. So really what we want to do is first just try to deeply characterize, phenotype this virus. And we've got 9 different panels of metabolites that we're looking at. And as I said, today we're going to stick with the lipoproteins and present the lipoprotein data, some of the inflammatory markers, the cytokine screens, and also the tryptophan kynurenine pathway and amino acids, because this is such an important pathway in a lot of the effects we see, from the neurological effects to the renal and the liver effects. So before we did any analysis, what we had to do was just have a look at our samples, check that they were good. And these 3 vector graphs show the effect of various pre-treatments. So freeze thaw, because we were sample limited, we had to do multiple freeze thaws for multiple mass spec assays. You can see those in the blue F1 to 5. This is showing up to 5 freeze thaws, and you can see it doesn't really pull the samples much outside of their origin after 5 freeze thaws. One of the other questions, more pertinently though, was, can we use heat inactivation of the virus, and can we use heated samples? And you can see the red vectors here. This does have a massive effect. So the answer we got was, no, we can't use the heat inactivated samples for metabolomics. I think it's not so bad for the proteomics, but certainly for the metabonomic analysis, it doesn't work. And was even more concerning was that if you heat the sample, it has a differential effect. Heating has a differential effect on healthy and the COVID samples. So we sort of got our sample protocols ready and then ran the samples. So we're having to work with samples that have been collected in the intensive care scenario. So the doctors are obviously more worried about saving patients than the quality of the samples, which is perfectly understandable. But we found that up to 24 hours of sat in a fridge, as long as you got the samples to a fridge within the first hour or so, that the samples were of reasonable quality. Now, the first thing you can see from the NMR data, if I just show you the healthy versus COVID, that's blue versus red, you can see that even looking at the NMR traces and not doing any supervised modeling, just letting the data tell their own story, the healthy are very, very clearly distinguished from the COVID positives. You can see some dark grey samples. These were hospitalized patients with respiratory disorders, but who didn't have COVID-19. And the green samples, the light green samples here, were actually our healthy controls. But what we found was at some point along the way, some of the controls we were using, which were mainly from our own laboratory and the hospital, were mapping with the COVID positives. So we then sent all the samples off for an antibody test. We looked at IgG and IgA. And what we found was that for several of the healthy controls from 40 actually had the antibodies, and these 7 people had all been to hotspots in the last 2 months and had some sort of cold, but hadn't got tested for COVID, didn't think they had COVID. But it's quite interesting that the most severe case of cold and somebody who'd been to Italy, Korea, and France skiing, in fact, all those people were mapping heavily with the COVID, and they had the antibodies. So that was quite an interesting find. Anyhow, when you look at the loadings to see what are the key differentiating molecules, the N-acetyl glycoproteins come up, GLIC-A, GLIC-B, they're sometimes referred to. And this wasn't surprising because this is a pro-inflammatory marker. You can also see a huge increase in glucose, again, not surprising given what we know about the association between the COVID and this disease. So looking at just the small molecules, the CPMG spectra, we trained the model on just 8 people, 8 COVID plus 8 controls, and we mapped everybody else in. And what you can see is that based on a training set of 7, which is a small 7 or 8, which is a small training set, all the COVID positives map correctly, all the healthy map correctly. And then you have the bottom left-hand corner, you have the grey patients, which were all COVID negative, either in the hospital or just turning up to a test centre. And then this group of, in green, we've got this group of controls who came back with the antibodies, and they're mapping mainly with healthy, but still in between the 2 groups. So the lipoprotein profile we measured 112 lipoprotein parameters, also showed a clear differentiation when you do the PLSDA. I'm showing the PCA here, but you can see that there's based on the lipoprotein profiles, there's a differentiation between the healthy and the controls. And in the bottom 2 graphs, what you can see is two patient trajectories. And if you look at the red asterisk, this is when the patient tested positive in hospital, positive here, time points 2 and 3. And by time point 5, they tested negative. However, this patient isn't going back towards the controls or healthy. It's moving, if anything, further into the COVID space. And the same with the second patient. So what this is telling you is that even although they have a PCR test saying they are recovered, there is the metabolic recovery there is no metabolic recovery at this stage. In fact, if anything, metabolically, they're more abnormal. And looking at the lipoprotein signatures, the serum signatures are very similar to carotid atherosclerosis. And you can see that based on the HDL, the LDL, and apolipoprotein B, and the apolipoprotein B to A1 subfraction ratio, you can see that these all match typical cardiovascular disease profiles. And we've got new studies coming out that show COVID-19 causes blood vessel damage. There have been several studies now. And if you also take those lipoproteins that are altered and you look at what happens in type 2 diabetes mellitus, everything with the yellow star in this graph is shared with COVID-19. So we see this increase in apolipoprotein B, we see an increase in VLDL fractions, TNF alpha is increased. We see decreases in the high density lipoproteins across all subcomponents, and also decrease in apolipoprotein A1. So quite a similar profile to type 2 diabetes. If we use the mass spec TQ assay for amino acids and biogenic amines, and here we've got on the right hand side, we've got increased in COVID positive, and on the left hand side is lower in COVID positive. What we see is that the COVID-19 is dominated by glutamate, kynurenine, phenylalanine, a lot of taurine, ethanolamine. These are all amino acids that we've typically associated with liver disease. On the left-hand side, you can see the disruption in the tryptophan pathway. There's less tryptophan and increased kynurenine. And we know that there's a role of kynurenine in COVID. There's several groups published that. But we also know that the kynurenine pathway is key in chronic diseases such as, but is also implicated in insulin resistance and diabetes. As I said, the decreased glutamate in the early phases of septic shock that you see in these COVID patients is reflecting the acute liver dysfunction, and it's been shown by some groups to be an independent predictor of patient survival. So this is an eruption plot. So it's basically plotting the Cliffs delta statistic with the OPLS loading. So anything in red and high up on either side of the graph is important. And on the left-hand side, we have the correlation with COVID. And on the right-hand side, we have the anti-correlation with COVID, so higher in controls. And just looking at this, what you see is the acetyl glycoproteins we're picking up from the NMR, telling us about the inflammatory story, which is backed up by some of the cytokines like CXCL10 and cytokine receptors like IL-1RA. But you can also see the amino acids coming into play here, elevated kynurenine and citrulline, and so you can see very clearly that it's not just one technology we're picking it up with, like the NMR with the glycoproteins and the lipoproteins, but also the amino acids from the TQ assay. So we come to the conclusion that it's the composite of the mass spec and the NMR together are giving us the clearest idea or clearest description of the COVID. And we're bringing people back, starting to bring people back in now into the clinics. And what we're finding is that the people who have the highest parameters here are the ones who are tending to have the persisting symptoms, particularly looking around the glucose control and the cardio effects. So going forward, what we're hoping to do, we have a population study which was collected 5 years ago, pre-COVID. So we have the Bustleton cohort, which is a baby boomer cohort. We've collected 4,000 of these people. And what we're going to do is use these guys as a control for the WA population going forward, looking at the COVID, and we'd like to think there wouldn't be a second wave, but we are still anticipating a second wave of infection. So we're hoping to be a bit more ready this time, and we've got all the procedures and protocols set up to collect samples coming through the hospitals. So we're going to be actively ongoing typing those. We're going to be bringing people back to look at the recovery. And then we've also, as I mentioned, got some samples from Cambridge University who are...
That's 2 minutes, Elaine.
Yes, okay, thank you. So we've got healthy people and health workers who've then contracted the disease, and we've followed them all the way through their either recovery or ITU journeys. So what we're doing currently is trying to set up a world kind of biobank of COVID samples and really try and deeply metabolically phenotype those. So just like to thank the rest of the team who've been working on this project, and thank you for listening.
Yeah.
Thanks very much, Elaine. That was great. A really interesting story, and you know, very timely, I think, to start looking into this. And so perhaps I could just start with a question or two. The patients, the positive patients in your initial analysis, and I appreciate that numbers were really quite low in those, but do they have comorbidities? So obviously we know that you know, obesity and diabetes are, you know, perhaps predispose people to the disease, but and particularly to poorer outcomes. And so have you got all the details of that in those patients, particularly compared to the controls?
Yes, so we did. We've got the controls and to the negatives, and what we've found, and also the Cambridge set, is that what we were seeing was obesity is probably the strongest associated factor. So BMI, and then hypertension we were finding. So we were surprised that asthma didn't seem to predispose. We had very few asthmatics, and not so many with overt heart disease or diabetes. I would have guessed that quite a few of the obese people were pre-diabetic, but maybe not formally diagnosed. But yeah, hypertension and obesity were the two main ones.
And so that really raises the question then, is the metabolic signature that you're seeing associated with the COP2, is that the result of them obtaining, of acquiring the disease, or was that a pre-existing and something that's predisposed them to the disease?
So we sort of looked at this, and if you saw in the principal components, the separation was in PC1. What you see in PC2 is the BMI effect and the diabetes effect. So BMI and diabetes sort of kind of co-map. So it's orthogonal but related somehow. And we've also looked at just the BMI and done the regression against all the parameters. And there's a couple of, you can see some of the parameters perhaps are shared, but the majority, particularly in the amino acids and kynurenine pathway, aren't pre-existing. This is definitely an effect of the infection.
Okay, that's great.
Thanks, Peter. The Q&A, will save the live Q&A till the end. Bal, do you want to pick it back up? Sorry, I'm being brutal with my timekeeping, Peter.
Okay, there was one question, but in fact that was similar to the question I asked, so I think you've covered that also, Elaine.
Okay, we're going to move to our lightning talks now. Just bear with me while I get that back up. Just for our attendees, you are all in listen only mode. So if you have any questions for our speakers, feel free to put them in the Q&A box and not the chat box, because we can only pick them up in the Q&A box. So if you post them there, then we'll keep track of the talks. So we have 5 lightning talks right now, and thanks again to Professor Elaine Holmes for that amazing keynote talk there. So our first lightning talk is delivered by Monique Ryan. She's going to talk to us about monitoring the inflammatory profile of acute burn wound healing in adults and the implications for improved burn wound management. Monique, I know you have 2 slides. Just let me know when you want to hit the next slide, as I'll control them. But
Thank you.
And you'll get a friendly alarm at 4 minutes 30, Monique.
Thank you very much. Hello everyone, thanks for the opportunity for letting me to present some of my preliminary data for my PhD project. So monitoring the inflammatory profile of acute burn wound healing in adults with implications for improved burn wound management came around because even though there's advances in burns treatment, there's still a lack of techniques for clinicians when it actually comes to monitoring that burn wound as it heals. For clinicians, it relies on constant observation of multiple parameters such as edema, the depth of the wound, any necrosis and so forth. And that also requires removing dressings as well, which could be detrimental to the healing process. Most of the research so far is only in severe burns that require a lot of attention, but there's only minimal research when it comes to acute burns with a total body surface area less than 15% burnt. So clinicians really need a quantitative marker that can predict the wound healing process 6 weeks or a month down the track without the removal of dressings and stopping treatments and so forth. So through metabolomics, we could identify key changes in plasma metabolites as the burn heals to potentially predict healing outcomes. And what we hope from this is that it can improve recovery times with guided treatments, reduce the economic burden of burns treatments, and also hospital readmissions for coming back for scars or any poor healing outcomes. So this study actually originally started with Fiona Stanley, which were collecting 61 acute burns patients over 6 weeks after they were burnt, and to progress and see how their wound was healing, as well as 22 healthy controls that didn't have any previous burns or problems associated with that. So with previously validated mass spec methods, we measured tryptophan catabolism metabolites and amino acids and amines using triple quads and a time-of-flight mass spectrometry from Waters and Brooker. And this is just preliminary results at the moment. We've only got a quarter of the way through the study. So we've done some multivariate and univariate analysis to identify any key metabolite changes. If I could have the next slide, please. Can I change the slide? Thank you. So we found some interesting findings in that from the 24 amino acids, we found 23 was significantly elevated in burns patients compared to healthy controls. And this was also seen with the opposite for the tryptophan catabolism pathway, where a majority of them were actually depleted in the plasma between burns and healthy controls. We also found a really significant finding in that serotonin levels in the burns patients significantly depleted 6 weeks after their burn was deemed healed. And so this might have a lot of implications in the long term, especially with immunosuppression and potentially abnormal wound healing later on with scarring and pigmentation issues. We also found through non-parametric testing that there was actually no difference in amino acid and tryptophan metabolites at admission for a burn and then 6 weeks post, sort of suggesting that there is a constant or persistent nutrient requirement for these patients and that potentially the burn has not been deemed healed after 6 weeks. And with the tryptophan catabolism pathway, it kind of indicates there is a disruption because there's the progression through the pathway is not being completed, and we think there might be issues with the tryptophan 2,3 dihydrogenase enzyme, which is responsible. It's the first step in the pathway converting tryptophan to chironine. So a lot more research will need to go through that. As this is only a quarter way through my PhD, we're going to do further supervised methods as well as pathway modeling for each of these patients because unfortunately our healthy controls are not age and sex matched. And we'll also compare them to the patient scars assessment scores for each of the 61 burns patients, just to see if there's any of these markers actually associated with poor outcomes. To address more about the inflammatory profile, we'll be having a look at lipidomic analysis to target inflammatory mediators and other lipids associated with that pathway to see if there is any changes to do with the inflammation and if they're associated with all of the factors that we found today. So thank you for listening. This is a lot of just preliminary data. We have a long way to go, but thank you for letting me present.
Thank you so much, Monique. So we're going to hold the questions. If you have questions, go ahead and post them in the Q&A box, and we'll take questions after all 5 speakers have presented their lightning talks. So our next speaker is, I guess, Xin Yi Wu from the National Taiwan University. And she's going to be presenting on the workflow of untargeted foodomic strategy for identifying fish freshness markers. Over to you, floor's yours.
Good afternoon, ladies and gentlemen. My name is Xin Yi Wu from Taiwan, and it's my pleasure to share with you our recent work relating to identifying fish freshness markers. And my first slide shows our workflow. Our first step is to select the marker candidates. We perform the comparative analysis utilizing the UPLC high-resolution mass spectrometry on the 3 batches of fish samples stored at 4 Celsius for 0 hour, 72 hours. And mass spec raw files were processed by MS style. And around 5,000 features were filtered out from each batch by setting the criteria of signal to noise ratio larger than 10. And the Manhattan U test with a significance threshold of alpha less than 0.005 was set between the 2 conditions, which resulted in 400 to 900 potentially changed features. And a total of 115 features were generated from overlapping features across 3 batches. And in our second step, targeted LC-MS-MS analysis was performed on these 115 features. And finally, a total of 16 metabolites were identified by using MS style coupled with MONA database. And 8 of them were considered as potential biomarkers since they passing the mapping and mass accuracy criteria. For example, the spectrum mapping should have the precursor and 2 product ions. And in the last verification step, the relationship between the response of these 8 potential indicators and the storage time for 0, 24, 48, and 72 hours was further verified by Spearman correlation. And all 8 markers have correlation coefficient larger than 0.7. Next slide, please. Next slide, please. Okay, thank you. And the 8 freshness indicators could be simply classified into 3 categories. The first group is the polyunsaturated fatty acid, including ALA, DHA, AA, and LA. And the second group is the medium chain acylcarnitine, including the decanoyl carnitine. And the third group is the degradation products of nucleotide, that is the uracil, hypercellin, and inosine. We finally proposed that during the storage, the enzymatic hydrolysis of fish lipids gives rise to the accumulation of free fatty acids. And with the stop in aerobic respiration, the fatty acid oxidation is rapidly redirected from mitochondria to peroxisomes, resulting in the reduction of decanoyl carnitine. And subsequently, ATP production stops and generates an abundance of several degradation compounds, such as the uracil, hypercellin, and inosine. So to sum up, our work provides a promising approach to assess the quality of fish during storage and gain deeper insights into the metabolic reaction. And thank you for your kind attention.
I don't even get to play my nice alarm. Well done.
Yeah, thank you so much. And apologies here to our speakers. I'm having a little bit of a time lag with when you say next slide, I am clicking next slide.
It's okay.
Apologies. Thank you so much for that
Thank
talk.
you.
Over to our next speaker. So our next speaker here is Georgia Sinclair, and she's going to be talking today about initial stress responses following exposure to contaminants. Georgia, the floor is yours.
Hello, fantastic. So my name is Georgia Sinclair. I'm a PhD candidate at RMIT University. My supervisors are Oliver Jones, Sarah Long, David Beale, and Paul Leahy. And my PhD research is interested in determining the initial stress response following exposure to contaminants, specifically PFAS. Poly and perfluoroalkyl substances are a group of over 4,700 compounds in a wide variety of that have been in use since the 1940s for a wide variety of commercial and industrial products, many of which I'm sure you have in your homes right now. Unfortunately though, today, PFAS has been detected in nearly every environmental compartment, so from your freshwater systems to your marine ecosystems, from your sediments to your soils. It's also been detected in organisms from macroinvertebrates to mammals. There's also been certain concentrations being detected in the bloodstream of humans and also in the breast milk of mothers. However, what's important to note here is that these concentrations are measured in nanograms per litre or micrograms per litre. From the ecotoxicology data that we have published today, they've seen that adverse effects on organisms that have been exposed to PFAS usually occur around milligrams per litre. So that's 1,000 times more than what's likely to be in the environment. So this concentration does not cause observable adverse effects on organisms that we know of. However, PFAS is a persistent chemical and it's unknown what the biochemical effects may be on an organism over time. So this is where my PhD comes into it. I'm wanting to know if we can use metabolomics to identify an early response to PFAS at low environmental doses in organisms in a range of organisms from aquatic to terrestrial. So be able to determine if we can see a difference between a metabolomic response when an organism is exposed through its diet or through water contamination. So as many of us know, metabolomics is the study of hundreds and thousands of small molecules in all living things, so your carbohydrates, your amino acids, and your sugars. They can provide us information on the internal functioning and the health of an organism, and environmental metabolomics can aid in our understanding of the interactions that organisms are having with their environment. It can also be useful to provide information on the toxicity of contaminants at a molecular level, so we can start to see those initial stress responses before we start to see adverse effects such as growth or mortality occur. Next slide, please. So my master's research, I contributed to the development of early warning bioindicators of pollution exposure using a metabolomics approach. So more specifically, investigating specific responses in an organism exposed to contaminants over time. So for this example, I used Simplicidia aquasatus. This is an estuarine sandworm found in the intertidal mudflats around estuaries around Victoria, Australia, and exposed them to two classes of chemicals, the heavy metal zinc and the fungicide boscalid. I exposed them to 10% of the LC50, so being 50% of the lethal concentration and 10% from that. We wanted to ensure that we were going to cause an effect, but not cause mortality. I then ran a time course exposure on these with the zinc exposure and the boscalid exposure for 2 weeks, collecting the samples at different time points and running a multi-platform metabolomics approach. So I used gas chromatography mass spectrometry to look at the entire profile of the organism and be able to identify specific metabolites or pathways that may be affected by the contaminants. We then used a more sensitive technique using liquid chromatography mass spectrometry, where we were able to target specific metabolites and be able to compare them across the different treatments. So in this case, we focused in on amine compounds. And so for the purposes of this talk, I'll just briefly explain to you what those results were. So we had 4 metabolites that significantly changed when the organisms were exposed to zinc, and they increased in abundance in the first few hours, and then at the 96-hour time point, they reduced quite dramatically. Whereas the boscalid, we had quite a number of different metabolites that significantly changed when they were exposed, all reducing in abundance from the 24-hour initial exposure point. So we had different metabolites changing from the different treatments and also changing over time, showing that initial stress response. So to summarize, we're able to use metabolomics to identify initial stress responses before we start to see adverse effects such as mortality occur. And we're interested in doing this using PFAS as our contaminants on organisms. This can then potentially provide another tool for diagnostics in environmental assessments and can also aid in setting guidelines for PFAS contamination in our environments today. And I'd just like to thank you all for listening.
Thank you, Georgia, for that fantastic overview. I really have an interest in PFAS and PFOS analysis, just given some of the challenges that we experience here in the Americas. So yeah, thanks for that overview. Our next speaker is Hui Yi. Hopefully I pronounced your name right. And the lightning talk is focused on decoding a glycolytic metabolite targetome in human cancer cell lines. The floor is yours.
Okay, thank you. Hi everyone, I'm Hui Yi from China Pharmaceutical University in Nanjing, China. So now I want to actually switch gear and talk a little bit about how we apply another powerful tool besides metabolomics. It's called chemical proteomics, and to study metabolism. So as we all know, a recent clinical omics study, as well as speakers from previous talk, have all shown us a close association between abnormal metabolism with disease incidence and disease progression. So actually these studies have thrown a question to us, is that whether the observed metabolic changes, are they just biomarkers or can they actually be game changers? So we believe metabolites, they are not just reflect changes occurring at the genome, transcriptome, and proteome level. They can actually possess certain feedback mechanism and send signals upstream to cells. So this hypothesis has actually been substantiated by several high-impact chemical proteomics studies. So by chemical proteomics, it's a technique developed to study what proteins bind to certain small molecule of interest. Anyway, these studies, they show that metabolites, they actually can bind to a multitude of proteins, and the metabolite protein interactions are able to affect the protein target's function by either directly changing conformation, altering PTM status, or affecting protein-protein interactions. So we believe the knowledge of the metabolites and what proteins they interact with, we term here as the targetome, is the key to understanding the mechanism of metabolic regulation. Next slide, please. Hello, next slide, please. So this previously has been achieved by derivatizing the metabolite of interest with either attaching them to solid phase or adding a click chemistry group, so we can pull down the proteins bound to the ligand of interest. However, any derivatization made to the metabolites risk in distorting the native metabolite protein interaction. Therefore, here we develop a novel accessibility profiling based approach called TRAP. So the rationale is simple. We incubate the metabolite with the cells, and then the metabolites will bind to its protein targets. Then we use a covalent modifier to derivatize all the reactive amino acids across the proteome level. So if the metabolite interact with its binding proteins, it'll induce direct hindrance to the covalent modifier, which means, simply speaking, the labeled peptide within the metabolite binding domain will show reduced intensity in the metabolite binding group compared to the metabolite free group. And then with quantitative proteomics, we can quickly assign what proteins bind to the metabolites of interest, and also determine the proximal domain the interaction occurs. So due to the time limit, I just talk very briefly about our results. So we benchmark the approach using a metabolite biphosphate fructose and an allosteric activator of pyruvate kinase. We find we can quickly assign the FBP binding site from PKM2. We also can discover the target protein for a small molecule drug, TAF46, directly from native cell media. We use 2 human cancer cell lines as well as E. coli, so we can even determine the TAF46 binding site from the complex cell lysates. Next slide, please. So then we set out to really apply the TRAP approach to study the targetome for bioactive metabolites. So because our group has special interest in cancer metabolism, we really are intrigued by the roles the glycolytic metabolites are playing in human cancer cells. Using TRAP, we indeed identify a multitude of target proteins from the acid.
30 seconds.
Okay. Anyway, we found the targetome constitute proteins of transcriptional activity. Some of them are previously defined as unligandable, exemplified here by TRIM20A and lactate. We validated the interaction between the two in both cells and using recombinant TRIM28. So the next question is where the interaction occurs and what biological consequences are implied by the interaction. We found the lactate affects the accessibility of the domain from recombinant TRIM20A at the domain responsible for p53 degradation. So using immunoblotting assay, we did validate the new transcriptional control mechanism for lactate. So basically lactate binds to TRIM28, which further promotes p53 degradation. It really shows us a new mechanism regarding why glycolysis is hyperactivated in human cancer cells, because it indeed provides survival advantages for human cancer cells in response to chemotherapy. So at last, I believe what we've seen is just the tip of an iceberg, and anyone who are interested in elucidating why metabolism is so powerful are welcome to join this field and uncover how metabolites interact with proteins.
That's time.
Yeah. And thank our group and the FIMS. So FIMS is a global initiative developed to support female scientists. It's also how I know Susie. Thank you, Brad. Sorry for running over time.
No, it's a worthy shout out at the end. It's okay.
No problem. Thank you so much for your talk.
Our
next speaker here is our fifth speaker of the Lightning Talks. So I want to introduce Cheka Kihalpanala. And what I'm going to do is I'm going to stop sharing because I believe you have an animation here, Cheka, and I'm going to hand the get you to share your slides, and then Brad can start the timer.
Got an itchy timer finger.
Right, thank you. Hi everybody, I'm Cheka from the Rosner Lab School of Biosciences, University of Melbourne. I'm going to talk about my project, a PhD project where I developed a tissue-specific lipid map of the model plant Arabidopsis across development. Lipids are highly complex, difficult to detect and identify. So to tackle these problems, I first developed an analytical workflow to detect and identify lipids from diverse Arabidopsis tissues throughout development using an untargeted lipidomic approach. I first compared 4 different commonly used lipid extraction methods, and the hot single extraction method was found to be the best to extract lipids from different tissues of Arabidopsis. A swath method using the SAG 6600 mass spectrometer coupled to 1290 HPLC was used to detect lipids, which were then identified using the internal lipid library of MS-DIAL software. Using this approach, we identified more than 800 lipids from 7 different Arabidopsis tissues, and in collaboration with the Provat Lab at the University of Toronto, Canada, we developed an EFP browser to display the data. This is the browser that we developed, and the lipid of interest can be selected from the drop-down menu, and its relative levels in different tissues and developmental stages can be visualized by the intensity of the color. Higher color intensity corresponds to higher lipid levels. Furthermore, by clicking on each of the tissues, the user can navigate to a page where the full lipid profile of that tissue is given, along with the characteristic fragments observed in the mass spectrometer. Even though significant changes were not observed for most of the predominant lipid species in Arabidopsis during development, many less common lipid species showed significant changes in their levels during development. We believe that our database will facilitate further investigations into Arabidopsis tissue lipids and their responses to developmental and tissue-specific cues. Thank you.
Thank you so much, Sheka. Okay, if our 5 lightning talk speakers want to turn on their cameras and come off mute, we're going to have a short Q&A round. And also to the organizers here, if you have questions for our speakers, feel free to jump in and take yourself off mute. I guess I just want to finish up by offering the questions to Elaine at the end of her keynote talk there. We did get a question from the audience asking what's the difference between PLSDA and OPLSDA?
Okay, so PLSDA, sorry, just shut the door, there seems to be a building construction going on there. PLSDA is a discriminant analysis where you tell the computer the class of the sample, and the algorithm tries to maximize the difference between 2 or more classes. With OPLSDA, that stands for orthogonal partial least discriminant analysis. The orthogonal part relates, so if you know what's related to class and you remove the systematic variance that is unrelated to class, you will end up with orthogonal dimensions. So for example, if you are looking at heart disease, and we know, for example, that gender is associated with heart disease, what you don't want to do is phenotype somebody based on being male or female. What the orthogonal PLSDA tends to do is remove the various factors, orthogonal 90-degree factors from the model, so that you get the orthogonal factors put into a separate matrix, and then you can then go and look at those. So basically, it's a filtering algorithm that cleans up your data. Like all discriminant analysis or supervised analyses, the more you do, the more you fiddle with the data or manipulate the data, the more prone they can be to overfitting. So it's very important to have a test set.
Thank you, Elaine. Hopefully, Hussein Ahmed, who put that question, that answers your question. That's really nice overview there. Any other questions from our organizers here? Peter, feel free, jump in for our speakers.
Yeah, thanks for that. Monique, I have a question for you. A very interesting talk, and I appreciate that it's early stages in your talk, so you haven't, there's a lot of things that you're going to do. But it was sort of interesting that you didn't see any effect over 6 weeks of time, and I would have expected that there would be some healing and the burn would be lessened over that period of time. And I wonder if you've got sort of an explanation for that. And the second point then, I guess, is whether you saw a correlation between the effect size in terms of the metabolites and the size of the burn, for example, or the severity of the burn.
Yeah. So I've done a little bit of data analysis into that way. So we do see that the metabolites do change a little bit over time, as it's we've got a time point, we've got 6 time points. Between on admission and 6 weeks later, the levels tend to stay the same, but we do see some peaks and drops of metabolites over time, and it regains to burns admissions. Don't exactly have a theory behind what is happening there. These patients do have a surgical intervention to remove their wound at around 2 to 3 days post their actual burn. So there could be that also factoring in that you also have additional trauma on top of just the burns. And when it comes to actually the TBSA, we do see a slight some changes in the metabolites, especially when we get up to 15 or 16% total burn surface area. We see an increase in the tryptophan, the amino acids, but anything below that, it tends to be, there's no real significant difference between them. Yeah, still a lot more to tease out there.
Okay, we have a question here from our audience. This is a question for Hui Yi. I really hope I am pronouncing your name right, and do tell me in all honesty if I'm not.
Yeah.
I have a question from the audience. Would all metabolites work well for chemical proteomics? Are there any metabolites that work better than others?
Okay, so it depends on which chemical proteomics approach you want to take. So if you want to do some juvatization-based approach, then the bigger the metabolite is, the better. Benjamin Kravitz, he has published a paper by juvatizing arachidonic acid, the long fatty acid chain with the click chemistry group. So for that kind of molecules, it's big. So the addition of a click chemistry group will not affect too much of the interaction. Also, if the metabolite you're studying is very chemically active, for example, it carries some functional group that's very active to bind to or covalent modify its proteins, then it's easier to fish out the targets. However, if you're dealing with something like us, like lactate and maybe glucose or other intermediates, then you certainly want to go with the label-free approach like what I presented today. So it's better to capture the weak and transient metabolite protein interactions. So from that angle, any metabolite can be probed. It's not, well, I hope it applies to all the metabolites of interest.
Okay, thank you so much. I'm going to park some of the questions here and save them for after the second keynote talk here. Feel free out to our attendees, continue posting your questions. Just let us know who the question is for, and we'll take questions and answers at the end of Professor Marcus Wenk's talk. So for now, thank you very much to our panelists and speakers. And I'm going to go ahead and share my screen again. And so I'd like to introduce our next keynote speaker. Would like to present Professor Marcus Wenk. He's the Provost Chair, Professor and Head in the Department of Biochemistry at the National University of Singapore. And he's the Director of the Singapore Lipidomics Incubator, or better known as SLINC. So I think for many of us as a lane, Professor Wenk doesn't need an introduction. Everybody knows him and his work and his group. However, he is a pioneer in the field of lipidomics and has been awarded a number of different awards, such as the NUS Research Recognition Award and the European Federation for Science and Technology of Lipids. He's currently the editor-in-chief of Encyclopedia on Lipidomics and the executive editor of Progress in Lipid Research. And so his keynote talk today is titled The Human Blood Plasma Lipidomics. So I will hand over to you, Marcus, and go ahead and share your screen here. Oh. I think we lost Marcus.
Has he gone? I think he might have clicked the wrong button. I better stop his timer then, huh?
Yeah, I'm really enjoying this, Brad.
Too much, maybe.
Too much, maybe. So, just for our audience, it looks like we accidentally have lost Professor Wenk. So we'll just give him a minute or two to come back. And actually, while we wait for him, Monique, you still have your camera on, so you're the lucky person to get another question. While we wait. So it's a question from Tu Zhang in the audience here today. Are your samples coming from patients with the same burn depth and the same mechanism of injury?
So no, this is actually quite spread out. We'll need to tease that apart as well to see if there's actually subgroups, but they do come from scalds, flames, chemical burns and radiation, so they do have different sources, and they also do come with varying depths as well. Of course, as I mentioned before, clinicians have to observe the wound depth. You can't really stick a ruler into a burn wound, so a lot of it is just based on what the clinician thinks, whether it's quite, partial, superficial, deep, and so forth. Luckily, with the total burn surface area, we have most of the patients within the 10% burn area, so they don't tend to have very big depth wound.
He was there.
Okay, thank you. Thank you so
Just
much.
see you're
for
on,
that.
Mike.
Yeah, to see your speakers on. I don't know if we have Professor Venk back. I don't see him here. Can we just check that he didn't join as an attendee? I'm just going to wait for our team to get him back.
I have a quick question for Cheka. How do you get access to that little visualization tool? Is that public or is it?
Not yet, not yet. It's
Okay.
almost done. Very soon.
Okay. Cool.
Yeah, if anybody else wants to come off mute from the panelists and pose questions to our speakers here while we just wait for Professor Venk to come back.
Yeah, Belle, just another quick question for Hui Yi, if you're still there. It was really a fascinating technology and technique that you described there. And I just wanted to try and draw you out a little bit in terms of what the potential is for the breadth of the proteome that you can screen with that type of approach.
Thank you, Peter, for the question. So actually, we are trying to develop it as a knowledge that applies to all small molecules. So actually, besides active metabolites that we found are associated with disease progression, we are also screening targets for drugs. Anyway, the purpose of this approach, besides elucidating the metabolic regulation mechanism, is really to identify functional proteins that are affected by metabolic changes. So we can develop a small molecule or just put like a drug lead compounds to modulate the target's function so we can cure or treat diseases. So that's the ultimate goal, and we are working on it by identifying certain protein targets. Now we found we can identify novel ligand structure that previously we don't know interaction can occur, and then we collaborate with organic synthesis chemists. So we design some compounds together to better control the activity of the functional targets that can control the disease progression.
Okay, thanks. Marcus, I see you've made it back.
Welcome back.
Well,
hey, sorry, folks. You've seen me in and out a bit. We've got some, contractors in the house. They are helping to fix something unexpected, and we told them from 1 to 1:30 I need a bit of no drilling. That they agreed to that, but when they left for lunch, they switched the power off, and so I lost all internet. Anyway, I don't know. Ready to go, or?
Yeah, please go ahead, Professor Venk. I gave your introductions, so the floor's yours, and I did stop Brad with his timer.
Yeah, I'll restart your timer.
So let me bring this back on, and thanks for having me here. Very nice this session. I'll join in for the other ones too. So here we go. Just 3 things from our end. I want to provide a bit of an update on our human plasma initiatives and our own work. So item 1 is really science, provide you a bit of unpublished data. Then 2, I want to make sure that you all know about these ring trials that we run. Many of you are part of it, but maybe a few others might find it interesting for the future. And then 3rd, upcoming events. So here we are. I mean, this is work just being submitted for publication, and it's a collaboration among different groups. There are 2 Marks here, Mark Chan and Mark Richards. They are both cardiologists, and they help with a cohort. In this case, these are 83 male and female volunteers of Chinese ethnicity who are asymptomatic for coronary artery disease, but they might, of course, come down as they age with some of these complications. They have a mean average age of about 57 years. And so Federico and Bo in our team then helped do lipidomics on these. This is targeted work. And you see roughly the numbers that we are covering here on this slide. And Hyun Wong Choi and his group, they help with statistics. And at the at this panel down here, you see the type of variabilities that we're getting into. And so I'm going to just guide you through this in the next couple of minutes. We really gotten a lot of interest in not only measuring between people, but also within individuals or within subject, as stated here. You can do this if you have multiple testing. So these individuals were tested every month over 5-6 months or so, and we have a circadian pattern as well for some of them. So that allows you to do a very different analysis to what maybe is done traditionally. And our motivation for this study really is twofold. We really have a keen interest and would like to determine draft lipidomic variabilities across the temporal domain in particular. But of course, our colleagues in cardiovascular, they want to check whether altered variabilities of such metabolites beyond cholesterol can be used to assess risk of disease, for which coronary scans are definitive. They provide a diagnosis, but if you had something that would provide you some early risk marker, then that would be of interest. And it's known, for instance, that increased biological variability of circulating cholesterol is associated with greater progress of disease. So here is the data, and what you can see in the bars are these so-called between subject variabilities. The lollipop icons are within subject. Just to bring this back to what I just introduced. A couple of observations here. First of all, maybe not entirely unexpected, but this is a first for us and for the field actually, that most lipids that are shown here, they exhibit lower within variation than between subject variation. So the lollipops are typically covered within the bar. Now there's a few maybe exceptions here with the phosphatidylethynolamines, where these values are about equal, and just a tiny handful, couple of serines, lysophospholipids, where it's the other way around. So where you would have larger within person variability as opposed to between. You can group them according to the overall spread. The glycerides, we've seen this before, they have the overall highest biological variability. I'd like to just emphasize that this is a draft of variability values that we now have here in this small cohort, but these type of values are not currently available for lipids, and we are very interested to study this further, and we really believe that knowledge of these variabilities will not only be of scientific interest, but also of value for testing, and in particular when done in combination with reference intervals, and I'll get back to that. So I don't have the time to go into all of these evaluations here, but I want to show you a few examples of what can be done with such data sets. So if you look at the top panel on the right, this is where we compare these variabilities, the within subject in particular, that's the one we're mostly interested here, to a risk score, a clinical risk score. The Framingham risk score, FRS, is widely used, and it has an input, the variables, age, sex, blood pressure, H and LDL cholesterol, that's then sort of computed into a risk, a 10-year risk for coronary heart disease. And so you could ask yourself, to what extent does our biochemical readout here that goes beyond the cholesterol level sort of, adds to that? And we see lipid association-wise quite little, actually. And so there's little difference in within subject variabilities between two groups. We have here the low risk and the medium to high risk, according to the clinical score. And we see 6 or so indistinct lipid molecules that have a slightly higher within person variability as opposed to the low risk. But these are small changes, maybe 1 to 2-fold, 1.2 or so, 1.3-fold changes in concentration. So, but that's just to introduce that we're mainly interested in understanding what does altered variability mean, be it higher or lower. I'm not making a point here whether it should be either way. It's about thinking about trajectories rather than anything else. And so let's move on to the next slide. This is a rather busy slide. I'm going to guide you through this, but common to all of the panels here is that these are all associations of some form of lipid level or concentration or variability with plaque burden. So here we take into consideration the angiography data, which is high resolution, so imaging, where the different categories of plaques were classified. And so panel A, for instance, is the comparison of single measurement. It's a bit like what you would do in a clinical setting. So you have a baseline measurement and then you take the risk from there, versus repeat measurements. And you see that the red and green dots, so that would be the single measurement readouts that overlap with the other one, are associated to some extent with a total plaque burden, but that you have a whole lot of a richer sort of picture if you do multiple testing. That's then shown in the purple icons here. B, for instance, is a different way of looking at the data. So that's more traditional, and many of us are familiar with this. So this is the lipid levels, concentration in a linear mixed effects model association with plaque burden. And so we find different patterns. Triacylglycerols associate with lipid-rich plaques. That's not unexpected, but we see a few distinct patterns for the other categories, such as the calcified plaques. And then this is maybe the key thing here for today. When you look into within subject variations, and that's panel C and D, we sort of classified them here into quartiles, low Q1 and high Q4 of lipid-rich plaques. So this is the one that the cardiologists care a lot about because the least stable, and so we have these low and high burden groups here, and ask ourselves, how do these within subject variabilities compare? So if you look at panel C, you find that the lysolipids they show up there quite prominently, and so we thought this could be interesting. Maybe this is a class-specific thing here. So we then tapped the third variability to move away from the population-based one, which is still the case for the within subject, into individuals. So now this is about comparing individual participants, not reference to anybody else over time. And so it's meant here phrased out, spelled out as visit to visit lipid variability is a common terminology also used by clinicians. So visit to visit as you go see your physician. And that's where we see these lysolipids come up very prominently as well, associated with lipid-rich plaques. So in summary, I mean, unlike the within subject variability, that is still the population-based sort of readout from the study group that you have, however big it is, the visit-to-visit variability is person-specific, and we feel that probably the one that over longitudinal readouts provides really a new type of data that will be very, very interesting. So I'd like to move on to briefly now touch on these ring trials shown here. The plasma ring trial, and that's a community effort really coordinated in the end now by the ILS, the International Lipidomics Society. Many of you are part of this. There's 2 trials currently ongoing, one on ceramides and another one on bile acids, and they both follow the same scheme. So we have a small coordination team that helps with this. spread out across the world, and also, you know, what is being done. So in both cases, we ship around to the individual labs. You see the spread here across the globe. The 1950 pooled plasma, and you know, it's a start. So that will help us understand a bit the technical replicatability across different labs, and we will certainly learn from this as what can be done as a focused extension to previous lipidomic you know, efforts in that respect, for instance by lipid maps and NIST. Now, so what you would receive if you're a participant is 1950 and appropriately titrated standards as a mix, and that stable isotopes that were put together by our additional partners that are shown here, that are coming into these trials. So the ceramides are rolling, and colleagues are returning results. We hope to provide an update later in this year. The bile acids, they are the next, the second example. They are moving now. There's a few more, so the ceramides are only 4. The bile acids, we're looking at 15 to 20 or so. So it's going to be a slightly more complex trial. And when you see at the top of the slide, it says phase 1. So this is really for us a more of a technical baseline effort. But, you know, there is an added benefit to this, as you'll see in a short while. Now, we're very confident that this will actually lead somewhere. And the reason why this is that if we're able to share a reference material, and the 1950 is a reasonable one, you know, we can discuss this further, but it is shown in yellow here. If we are, as a community, placing a shared material, that means we have the same, and so it should be something that is easily available along our analytical batches and queues, on top of our own QCs that we might all have. These could be long-term references or other things, you know, interspersed with our unknowns and samples. Then these shared reference material QCs, they're going to be an outstanding tool to, you know, normalize. And that's what's shown in this slide. This is one figure of a recent publication. This is a long-standing collaboration also with Peter Mickel and his team, where we've compared you know, replication across different laboratories. That's on the left side. So site 1 could be us, and site 2 could be Peter's group in Melbourne, where we look at data before normalizing to a shared reference material and afterwards. So you see this brings it together quite well. You have it on logarithmic and linear scale here, but it also can be extended to different platforms. And that's the right panel, where you see direct infusion high resolution mass spec on the horizontal and reversed phase MRM targeted on the vertical. And you see even in that case, a very different type of platforms, you can bring the data together better. So we're confident about the approach, and that this eventually can lead to better comparability of such plasma lipidomics in the community. Now, you know, these reference values, I just want to briefly mention this here. They are important, and maybe the most commonly used decision support tool for interpretation of test data. So that means, you know, somebody would go into a bracket and say, where do I sit with my test result with respect to the lower and higher intervals determined for a particular population, and widely used in the clinical community. Unfortunately, we don't have any of that for the chemical space that we're interested in, and we feel that this effort, these ring trials, can contribute to that. So they will help a lot in our engagement with clinicians. So in the next phase, and we're already thinking about this, we want to move beyond, you know, analyzing pooled plasma, of course. This is not only about standardizing, but this is about getting to these reference ranges or intervals in different populations. And I hope many of you, as you're contributing to this, will remain committed to these efforts. That will then allow us to go back to this network, grow it a bit further, but then measure populations that you all have via your local medical colleagues and collaborators that we can then easily, extend from our technical trials into biological variation in different populations. We have ethnicity in Singapore. Peter is interested in cardiovascular, and so on and so forth.
Another minute, Marcus.
Yeah, that is my last slide. Thanks a lot, Brad. Upcoming events. We've started our own small forum in lipidomics, which is thematic and ad hoc. In September on plasmalogens. This was quite an interesting scientific lineup of recent progress in that sphere. We'll update in December, I just mentioned that, on the plasma initiatives with more detail, both the ceramides and bile acids. Please reach out to any of the names you've seen or myself if you're interested in learning more about this. We're glad to provide more info. And watch out for announcements on the 7 to 11 at Sling. There's going to be more to come as we move into the next year. I don't think we might be able to meet soon in person, unfortunately. But we have our international symposium in March coming back. In addition to being a scientific symposium, it will also be a virtual exhibition that will allow vendors to showcase their products a bit more and reach out to all of us using the technologies that are now being used for such exhibitions. Well, with that, I'd like to end here. Thank you very much for having me on this, and I look forward to discussions over the next month. Thank you very much.
Perfect. Thank you so much, Marcus. We have some questions for you here already. And then we'll open up the floor to the rest of our panelists and to Elaine as well to join for other questions that have been posted while you were talking. So here, a bit provocative, very interesting talk. Are you positive that you can use lipidomics to find reliable biomarkers?
Oh, well, that's what we do, but you might argue that other metabolites carry more information, right? So, I mean, lipids are tightly connected with a lot of different aspects of human physiology. So I believe that having a good readout on those will tell you something, but yeah, I mean, we'll see to what extent it can go beyond the cholesterol type of markers for which lipids are maybe a prime example, right?
Sure. Next. Yeah.
Now, could I ask a follow-up question to that?
Go ahead. Yeah, please.
Yeah, Marcus, and I think that was sort of relating. I was quite surprised as well that the level of intra variability within individuals was really quite high and higher than I would have expected. And, for many of those, it seems to be as much as there is between individuals. And so there's a couple of points that come from that. And that I guess raises the question of how well can we use lipids as biomarkers if the variation within an individual is as great as it is between individuals, then obviously trying to use them to differentiate individuals with different risk of whatever will be difficult. And sort of my follow-up to that is, have you looked at this data to see whether the variation within individuals goes in modules, if you like? So if do you see a number of lipids showing the same type of variation within individuals? So they go up at one point and then come down, indicating that there might be changes in metabolic pathways that are corresponding to these changes, and that's within an individual over time.
Yeah. No, we're not. So this is the first type of analysis that we're running here on this data. There will be more as we're looking deeper into this, and this is 5 months, right? So but you can, for instance, if you just look at these individual patterns, you can create some form of personalized, plot based on this data that would give you maybe an identity of an individual based on the lipidomic 5 months history that you have. So I think these variabilities, actually all of most of the within person variabilities are below the between group, and that's really what you would see also for many other metabolites out there, be it small molecules or salts, right? And so And so, a long term, what we would like to do here is to provide these dimensions, particularly within subject variability, to a growing list that the European Federation for Lab Medicine has recently taken over to curate this data together with the clinical details that they have there. These variabilities, I think, will be very, very important for testing in the future as we move towards more individualized readouts in connection also to the reference intervals. So if somebody spreads the entire range of a reference interval, for instance, that would have different implications for how you can use such a marker for screens.
Mm.
Along those same lines, there's a question here from the audience, Marcus, around technical variability. So how does that vary versus the subject variability that you've shown here?
Yeah, that's right. So our tech variability, we have that information here as well, of course, and it's tabulated for each individual metabolite here separately. That's taken from these QCs that I mentioned. About 1/3 of the samples that we have are QC samples. So if we have, this information is there. On average, if you add them all up here along the wheel, it's about 12%.
That's pretty impressive, yeah. We've got another question here from the audience. Actually, I'll jump to there's some posted in the chat box. Hussein asks, what software have you been using to process your lipidomics data?
Yeah, so that it's a good question. Thanks for asking that. We are short of software, but the person that has really become very active here, so Bo is our internal data chief, and he makes sure that this is all put together appropriately with the necessary QC material. And then it goes on to Hyun-Wong Choi, so he's become a member of our extended program. He's a stats team and a stats professor, and he and his group, they move forward. So they're looking into the statistics of the variability, but more recently also, and that's just been published in Analytical Chemistry, come up with a automation to do integrations, for instance. I mean, it's called MRM Kit. It allows you to deconvolute quite well, actually, large numbers of samples. You see, I mentioned here 83, but many of the other cohorts that we measure are by the thousands. So you're absolutely right. The software is a big bottleneck for us. So Hyun-Wong is a good contact, and I'm glad to help with that if you have specific queries on aspects of software.
Great, fantastic. Maybe we'll take another question here from the audience. Actually, from our next keynote speaker for next Friday, from Neera Kokani. Great talk, Marcus. Yes. Ring trials are critical for identifying variation in biological range in different populations. Would be interesting to see the contribution of lipid QTLs to this variation. Is there a plan to collect the genetics data?
Yeah, that's a good question. We should talk more about this, right? Yeah, you see, thanks, Neerja, for bringing this up. The two phases of the ring trial, I wanted to make sure that the growing community is fully aware of this, but we haven't really got our other thoughts around how to extend and connect this better to, yeah, exactly, genetics or so, which we might do as part of the precision medicine initiative. So thanks for highlighting that.
I'd like to open it up to the other panelists. Actually, are there any other questions from our organizers for Marcus? We got through most of the ones here.
Yeah, well, yes, but I have one more question, Marcus. Very nice talk. Thank you. And this regarding the variability within visit to visit, is that depend on the diets that the patient's having, or is there any relation?
So that's a good point. I mean, the diet is it's always a shortcoming. I mean, and these participants, they've been made aware of this study and asked to not to start the diet after the second visit, but you have no control over this. So there might have a slight fluctuations there, and we don't have much information on this other than the standard questionnaire maybe, but we there's no control. So we can't, you know, we have to accept this as noise. Absolutely.
I have a question, Marcus, or two maybe. One is mainly around the ring trials. Just interested in why the ceramides were chosen out of interest to begin your ring trials with.
Yeah, so the ceramides were chosen as part of a workshop here in Singapore a couple of years ago, where a small group thought ceramides could be of interest because of various reasons. I mean, they're measurable, we've got standards, the clinical community is increasingly interested. There is a test, a single, you know, measurement test that is out there using exactly these 4 ceramides that we're measuring for cardiac applications, so that these could be a good group to go after. But by no means does it sort of disqualify any of the others. And but this was a reflection early on, you know, so practicalities and interest for translation.
Perfect, thank you. And then the follow-on question actually was around the figure you showed, where I think it's really interesting that you can really standardize lipidomics data coming from various different platforms. Is there a goal to, for example, pull protocols together around best practices and share that with, let's say, more of the non-expert lipid community, and making lipidomics more accessible outside of gurus?
Absolutely, it's a good point, and as a matter of a lot of discussion. I mean, many of you are part of this, and you know we've spoke about that. So in this trial here, for instance, we provide what we call a standard protocol in both cases for the ceramides, and we will also do that for the bile acids. And they're up for people to adopt. We don't want to be in any way prescriptive. So if somebody would like to come on and say, I'm not doing ceramides, so a bit of help with the protocol, then that person could choose to use that one. But everybody is free.
I think we lost Marcus there for a little bit. We'll wait for him to come back. In the interest of time, when he does, we'll get him to finish answering that question. And yeah, we can move on to questions to the rest of our presenters. And Kino, I have a question here for you, Elaine, if you're still on the, I see you're online still. Thank you for bearing with us there. Are you able to tell, this is a question from Oliver Jones, a friend and colleague. Are you able to tell if the after effects of COVID-19 are a general post-viral fatigue, which is a common response to many virus infections, or is there a distinct, or if there a distinct effect of SARS-CoV-2?
So that one's a little bit difficult in that, bearing in mind we haven't looked, you know, we're looked at hundreds rather than thousands of samples, but it looks like there are a lot of common things that all add up to make it specific. So by that I mean, we still see disruption in the kynurenine pathway. We see a signature that's more like type 1 than type 2 diabetes persisting in some people. The acetyl-glycoproteins, which is kind of general inflammation, still up. And some of the amino acids that are indicating renal perturbation, again, still up. The lipid profile still. So if you took any disease like cardiovascular disease, you could say, oh yes, the lipids kind of match that. But then if you look at the amino acids, you're still seeing the liver effects. So it goes beyond, I think, the general post-viral stress, although we do see that signature too.
Okay, perfect. Thank you so much. I'll come back to you, Elaine. I've got another question here, but I see we've got Marcus back. So Marcus, you're on mute. It would be great if you could just finish off that last thought that you had around sharing of protocols and best practices. Oh, Marcus, you're on mute.
lipid maps and ILS, they are very active as well along very similar lines, and they go further into maybe, you know, standardization and best practices when it comes to what should be done for analysis of a particular class of compound. So they are submitting and providing information on guidelines papers beyond what I showed here in a very practical application. So look out for those as well. So that's Valerie has been quite active, and Kim and Gerhard.
That's great. Thank you so much. Maybe we can share some of those references with over the next couple of sessions.
Yes, absolutely.
Yeah, thank you. Okay, we'll go back to Elaine. I have one last question here for you. Is it possible to tell what changes were caused by COVID and what changes were caused by the immune cells?
So we haven't actually, we've done some analysis on the new immune cells in collaboration with Cambridge, but we haven't actually looked at those data yet. They have a very detailed cell-by-cell profile of the immune cells, but yeah, basically we haven't got that far yet. Okay,
thank you. Yeah, I'll just open up the floor now for any questions for any of the other presenters from the organizers.
Well, could I follow up a question for Elaine, and perhaps more a discussion point? Elaine, you know, I think clearly in the case of COVID, the current limitation is really around samples and you know, patient phenotyping and follow-up and all of those sorts of things. I mean, in Australia, we're severely limited, fortunately, I would say. But I spoke with Jeremy a while ago, and certainly I know you guys were sort of trying to recruit from overseas and so on. And so I think, in fact, this forum is also a bit of an opportunity to put out a call for people who may have access to cohorts, but how we might bring those together so that we actually have, you know, the numbers that we really need, I think, to really tackle and dig deeply into the metabolome and the lipidome of COVID patients. So have you had any success in that area?
We have. I mean, it's mainly been Jeremy Nicholson that's been driving it, but we have collaborations with Malaysia, with Cambridge, Boston, Spain, Baiguna. So there are quite a few collaborations, and the idea is to build up a composite database of well phenotype, and potentially some groups in Brazil. So yeah, you're absolutely right. There is a need to, and Jeremy's actually gone part way to setting up a network. So I think probably getting in contact into that. The problem's been very much that, when you get the samples, it's sort of the dregs. So people want to give you the samples to do the analysis, but there's sort of like, a couple of hundred micrograms, if that. So it hasn't always been the easiest to standardize assays because we've had to adjust based on sample size. So I think that's one thing when you go into a consortium that we need to be aware of, maybe during this second wave, that's happening in a lot of countries now. We can get some better protocols and better sample quality.
Yeah, people might be more prepared this time around, I think, yeah.
Yes.
I believe we are at the end of the Q&A session. We did run 15 minutes over there, so we do apologize, but the talks were phenomenal, and we got some great questions. I guess what I'd like to do is thank our speakers for today's presentations, and especially to our two keynote speakers. And I just want to go ahead and share the agenda for next Friday's virtual podium event. Another two great keynote speakers here, Dr. Karnani and Professor Masanori Arita, and some great lineup here for the lightning talks. And can you take the challenge and stick to a 5-minute lightning talk? So thank you so much to everybody. And yeah, as spooky as it sounds, it's Friday the 13th, but we'll see you here at the same time. And thanks again to our presenters and the attendees.
Yes, thank you to everyone. You made my job easy.
Thank you so much.